Okay, hi everyone. I'm Jeff Johnston. I work in the Genome Center as a bioinformatics scientist. Is it not on, or am I just not close? Is that better? Um, okay, so just a quick overview of what I'm going to talk about. I'm going to do a little introduction. It's going to be very biology 101, so I apologize for many of you. If you have any memory of your high school and college biology classes, you might get a little bored, but it's important for where we're going. So I'm going to talk about all the different kinds of RNA that are inside cells, and then a little bit about how we end up sequencing it for RNA-seq. I have a short few slides on a clinical application of RNA-seq, which unfortunately is not super common, but it's a cool example that Neil told me about. And then I'm going to spend some time talking about sort of the design and interpretation of RNA-seq experiments if you were sort of interested in doing this as a research project. And then I just have a quick list for you of RNA-seq resources that might come in handy. Okay, so introduction. You probably remember this from high school biology. It's called the central dogma of molecular biology. It says that information in DNA is transcribed into RNA, and then RNA is translated into proteins. Like pretty much everything you learn about biology in school, this is like a dramatic oversimplification. And it kind of doesn't give you a full appreciation of RNA because it makes it seem like RNA is just an intermediate between the DNA and the protein, which is not at all the case. So a slightly more sophisticated version of the central dogma is more about how information flows between these biomolecules. We all know that DNA can be transcribed to RNA, and RNA can be transcribed or translated into proteins, but there are other processes as well. For example, DNA replicates itself, so that's when a copy of DNA is made from a DNA template. RNA can actually also replicate, like viruses will replicate their viral genomes if they're stored in RNA. And some viruses can actually convert their RNA into a DNA template, so that's called reverse transcription. But notice that there's no known ability for, say, the information in a protein to be converted back into DNA or back into an RNA. So you all know that you just have one or two copies of your DNA in all your cells, and that's pretty much identical except for whatever somatic mutation you've managed to acquire. So RNA is a lot different. It would be very wasteful for all your cells to transcribe all of the RNA and produce all these proteins because proteins are very cell type specific. And in fact, the RNA profile of a cell, it will change during development because lots of very elaborate gene regulatory networks are active in development in order to get you from an embryo into an adult human. The RNA in a cell is also extremely cell type specific. This is sort of obvious because proteins, you might not need, say, enzymes for metabolizing drugs. You don't need those like in your skin. That's more something that's found in your liver. And like, say, neurotransmitters, you only need to produce those in your brain, not in your other cell types. So the other thing is that RNA is very dynamic, and it can rapidly respond to the environment. There are sort of these signal transduction pathways that allow the cell to respond to extracellular signals and trigger the expression of genes. So the fourth point here is that the RNA in your cells is not always translated into proteins. In fact, the vast majority of the RNA in your cells never becomes proteins. So that is a nice segue into the many different kinds of RNA. We generally group RNA molecules into two sort of families. So the first is this coding RNA. So this is the RNA that's eventually going to become proteins. We call it mRNA or messenger RNA. And then there is a large number of other molecules of RNA, which we sort of group into this non-coding category, which I'll talk about in a minute. But just to give you a quick review on messenger RNA. So the DNA template is essentially directly copied into RNA, into what we call a pre-mRNA molecule. This pre-mRNA molecule is then processed by the splicing machinery of the cell to eliminate, oh, the trackpad is also advancing. These pre-mRNA molecules are processed by the splicing machinery of the cell to remove these introns. So the introns are basically DNA sequences that are not used to code for the protein. So they have to be cut out in order to produce the final messenger RNA. All right. So, however, it's a little more complicated than this because the cell can choose to splice out more than just introns. It can actually splice out other exons. So if you see in this example, the same pre-mRNA molecule was spliced two different ways. In one way, it just got rid of the two introns, leaving 3 exons. But in another case, an intron, 2 introns and an exon were spliced out. This created a shorter mRNA. So we call these two different mRNAs transcripts. And this whole process is called alternative splicing. This is where a single gene encoded in DNA produces multiple different mRNA products called transcripts. This sort of dramatically increases the complexity of the transcriptome because now the cell not only can decide which genes to express, but can also express very specific versions of these genes. These transcripts, because they have different exons, they're going to produce different proteins. And some of these exons sort of endow the protein with different capabilities. So maybe if we think about, let's say exon 2 codes for something called a DNA binding domain. This is basically a property of the protein that allows it to seek out and bind to specific sequences of DNA. In the absence of that DNA binding domain, the shorter mRNA transcript is going to need help to find DNA, whereas the longer version on top is just going to sort of automatically be able to bind to DNA based on that sequence recognition. So that's just one example of how the cell can produce very similar proteins that have slightly different capabilities using this alternative splicing. All right. So as I mentioned, the majority of the RNA in your cells is actually not turned into proteins. It belongs to this whole class of non-coding RNAs. And I'm just going to talk about a few of these. I would be willing to bet money that if we opened up Wikipedia and went to the non-coding RNA site, we could find an example of a class of non-coding RNA that no one in this room has ever heard of. Because there are literally hundreds of different kinds of this non-coding RNA, and we do not understand most of it. So the major kind, in fact, probably about 90% of the RNA in your cells is made up of this translation regulation RNAs called rRNAs. And then the tRNAs are the transfer RNAs that are responsible for helping to build proteins. So in addition to that, there are these link RNAs. This stands for long non-coding RNA. These are involved in regulating transcription itself. These small nuclear RNAs, snRNAs, these are involved in splicing. And there's two classes of short RNAs called microRNAs and small interfering RNAs, siRNAs. And these also regulate genes, but they usually do that by binding to mRNAs and somehow affecting their translation. And then there is another group called piRNAs that stands for Pee-wee interacting RNAs, and these are involved in transposon defense. So basically, the genomes of almost every animal are sort of infected with these transposons. These are selfish DNA elements that will, if left to their own devices, essentially copy themselves randomly throughout your genome, which would be very, very bad. So animals have evolved this very sophisticated transposon defense system involving piRNAs and then this Pee-wee complex. All right, any questions so far? This is just sort of a small taste of the complexity of RNAs. All right, so, sorry. I think Emily covered a bit about how RNA is sequenced. So if you just, I always feel bad showing slides like this because I have never actually done this procedure, but I'm pretty sure it's not just 5 steps. But basically, you have to collect a tissue sample, and I just talked to Emily about this, so we typically need millions of cells to go into one of these RNA-seq experiments. So that's a decent amount. There's an RNA extraction procedure. So remember I mentioned that RNA is very dynamic. That means like the time between collecting your tissue and extracting the RNA can actually have a big impact on your experiment because these cells can sort of respond to this trauma of being extracted. So generally speaking, you want to minimize the time between when you get your RNA and when you actually extract it and produce your library. All right, so next you have to decide which RNA molecules you want to sequence. Like I mentioned, like 90% of the RNA in your cell is this ribosomal RNA, so you generally don't care about that. So a lot of these kits will help you filter that out. And then you can decide whether you just want the messenger mRNA or messenger RNA or total RNA, which would include some of this non-coding RNA if you happen to be interested in that. And then there's also kits that specifically focus on very short RNAs, like this microRNA and piRNA that I talked about. So sequencing RNA, it's kind of a misnomer because we don't actually sequence RNA. I don't even know if we know how to without these sort of more advanced single molecule sequencers. So what we do is we use this enzyme called reverse transcriptase to convert it to DNA. This is typically referred to as cDNA, meaning complementary DNA. That is an indicator that it came from an RNA template. And once it's been converted into DNA, we can just treat it like, you know, an exome sequence or a whole genome sequence. We just throw it on the sequencer and we get back reads, just like you guys have learned about this week. So what can we find out when we sequence this RNA? So the most obvious thing is we get to see which genes are being expressed because their mRNA molecules are going to be floating around in the cell and we're going to capture them and sequence them. But we can also see sort of more interesting things, like for example, if you have a genetic variant that would show up in the mRNA, you could see whether both versions of the variant, if you're heterozygous, for example, if both versions show up in the mRNA. So that would tell you whether one particular copy of the gene is not being expressed, but the other one is. You can also discover which transcripts of a gene are expressed. I'm going to show you an example of that because it's a little more complicated than you'd think. But basically, by looking at which exons your reads are arriving at, you can sort of deduce whether certain transcripts are expressed or not. You can also see basically deficiencies in splicing. So if you were to sequence mRNA and you started seeing a lot of introns in your mRNA, you could conclude that this, for some reason the cell was not efficiently splicing some of these molecules. In particular interest for cancer are gene fusions. This is when some sort of chromosomal rearrangement has basically brought together two parts of two different genes, and then that fusion is then expressed as its own mRNA. So this is can be somewhat reliably detected by RNA-seq. There's some sort of gotchas, of course. And you can also, let's say you don't have the DNA for somebody, if you do an RNA sequencing, you can see genetic variants because obviously the RNA is just a use the DNA as a template. So whatever variants were present in the DNA will also show up in the RNA. There's also a process by which cells can actually modify the bases of RNA after they've been transcribed. This is called RNA editing. It has I'm not sure I know a good example of RNA editing, but I'm sure if you ask me, I can find out one. Okay, so the real power of RNA-seq comes when you do comparisons between two different conditions. This allows you to see just what changed in the RNA. For example, if you have a cell line, you could expose some of the cell line to a drug and have a control as another set of cell line, compare them and see which genes went up and down. You can also compare tissues over developmental time, like say a child liver versus an adult liver. You can look at cancer. You could, if you do a biopsy of a tumor, you can also do a biopsy of the nearby normal tissue and compare the two. You could look at what happens when a pathogen infects a cell, maybe infect a cell line with a virus and then compare it to the uninfected cell line. So when you do these comparisons, you get a lot of really interesting information. So of course you're going to get the differences in genes themselves, but you can also get stuff about like which non-coding RNAs have changed in expression in response to your two conditions. You can see whether different transcripts of the same gene have been expressed in response to these treatments. And you can also monitor, say, if there's some sort of splicing deficiency in response to your treatment or a drug or whatever you've done. Okay, so you guys have all seen IGV with whole genome data or exome data. So this is an example of what an RNA-seq experiment would look like if you opened it up in IGV and looked at the reads. Down at the bottom here is the gene model, and these thicker portions are the exons and a different section of the mRNA called untranslated regions. And then the tiny line are the introns. So if you look at these reads, you can it's pretty obvious that the introns are basically missing. And that's because they've been spliced out by the cell, and we've sequenced the mRNA. Any questions so far? So this gene is very simple. It's just basically these two introns. I'm going to show you this more complex gene, which is kind of more a realistic idea of what you might see. So this is a very famous gene, TP53. It has many different transcripts. And in fact, just to sort of see this level of detail, I had to zoom in quite a bit. So all these longer transcripts are actually going off the screen to the right. But if you kind of line up the exons of these different transcripts and their reads, you can kind of see how we could deduce which transcripts are expressed in this situation. For example, I don't know if you can see the pointer, but these small exons right here, we don't get any reads over them. So that means right off the bat, this is driving me crazy, Neil.
Pointer.
This?
Yeah.
It works?
Oh, yeah.
Okay.
Green button. Try that. Yeah, there you go. That guy.
Oh, okay. That is much better.
Way less.
Yeah, so right off the bat, we can basically eliminate all these transcripts. Now you can't hear me, can you? Sorry. All right. Oh, it's brilliant. No, I'm not going to do that. So we can automatically eliminate some of these transcripts just because we don't see any expression over these exons here. And if you look over here, these shorter transcripts, we can eliminate them because we don't actually see enough reads over this longer UTR portion. So this is kind of what the software is going to do behind the scenes. It's going to analyze all these reads, and it's going to make its best guess as to which of these transcripts are the one that are being expressed. And it's important to note that it doesn't just have to be one of these transcripts. It can actually be a combination expressed in different proportions. Yes? Is there significance to the white arrows on the first exon? The white? Well, the arrows are just an indication of that this is on the negative strand. The gene is on the negative strand.
I'm just curious why there's some of them and not the other UTRs.
Oh, yeah, yeah. Okay, I see. Yeah, yeah.
Okay.
Right. Does that make sense? Yeah. Okay. So that was a quick introduction of how you would, what RNA-seq would look like in the browser. I'm going to just talk about this case that Neil told me about. So about 3 years ago, not quite 3 years ago, this paper was published about using RNA-seq to diagnose muscular dystrophy. So muscular dystrophy is generally caused by this particular type of muscular dystrophy is caused by a mutation in this DMD gene, which I've shown here. I don't know if you can read that, but this gene is extremely long. This is almost over 2.2 megabases, and it has many different transcripts. So in this case, I think there was like an 8-year-old boy who had all the symptoms of muscular dystrophy. So they went ahead and they sequenced the DMD gene, the exons of this gene, basically equivalent to exome sequencing. And they did not detect any causative mutations that would sort of disrupt this gene. They went ahead and sequenced other genes that are associated with muscular dystrophy, and they also did not find anything that would sort of explain this kid's symptoms. And there's also an assay that allows you to sort of detect the presence of DMD protein in muscle cells. And when they did this assay, this protein was essentially absent from this kid's muscle, which is a very good sign that something is disrupting the expression of this gene. So they did an RNA-seq experiment, and this blue color is the patient. This is the RNA-seq of the patient, and there are 3 controls above it. And they saw this very interesting thing, which is, for some reason, it was as if there was this extra unannotated exon appearing in this kid's RNA for this DMD gene. This is like the full gene, and this is just like a zoomed-in version. This is their paper. I apologize for the numbers. So you probably can't read this, but these numbers indicate like the number of reads, normalized number of reads, and in the kid's case, it was like 8-fold lower than the control samples. So definitely a sign that something was not quite right with the expression of this gene. So what they ended up finding was based on the sequence of the RNA, they could identify a SNP inside the intron of the DMD gene, one of the introns of the DMD gene. And this intron was essentially creating an exon. So the cell recognizes intron-exon boundaries using specific sequences, as well as some other rules that we don't sort of fully understand. But in this case, it was essentially convincing the cell when it was splicing this mRNA to create an exon that shouldn't have been there. And this exon just happened to code for a stop codon in the middle of it. And typically when a cell detects an mRNA with sort of a premature stop codon, it goes through this degradation pathway. So all the transcripts for this protein were basically being degraded by the cell immediately after they were transcribed, which essentially explained, basically had a genetic explanation for this kid's muscular dystrophy. So that paper is open access. I think you guys have access to these slides eventually, so if you just type in the name of the paper in Google, you can read all about it. Okay, so the next part is about the design and interpretation of RNA-seq experiments. If you do an RNA-seq experiment on, say, a single sample, what will happen is it will get processed by a software tool called an RNA-seq quantification tool. Its job is to look at all these reads and estimate an abundance for each transcript. You can actually do this at the transcript level or the gene level. The gene level is really just collapsing all the transcripts into a single value. Most researchers prefer the gene level because unless they have a specific knowledge about how the different transcripts of, say, a gene that they're interested in behave, they don't really know what to make of all these transcript information. So you'll either get a list of transcripts or a list of genes, but they're effectively the same. So you get this abundance measurement. And this is sort of, I feel like, one of the most confusing things when I talk to people about RNA-seq is sort of what this abundance measurement actually means and how you can sort of interpret it biologically. So fundamentally, the abundance measurement is just telling you what portion of the transcripts that were observed in the RNA-seq library belong to this particular transcript. The idea is that if two mRNA transcripts have the exact same number of copies, they should have the same abundance value. And that should be regardless of how long the transcripts are. And sort of what follows is that if you were to have an mRNA that has twice the number of transcripts, then it should have twice the abundance value in your RNA-seq experiment. So it's important to notice that I said of the total number of transcripts and not sequencing reads. And that is because transcripts have different lengths. And so if you don't normalize by the lengths, you could have a gene with the same number of same number of copies, but if it's twice as long as, let's say, gene A is twice as long as gene B, just by default, you're going to get twice as many reads from the longer gene. So these RNA-seq quantification tools go through a lot of trouble to try to normalize this length. And this is actually very sophisticated, and I feel like every other week a new RNA-seq analysis tool comes out. But the other thing to keep in mind is that even though the mRNAs are the result of transcription, they don't necessarily reflect the rate of transcription for these genes. And even though mRNA molecules are translated, you can't tell from the RNA abundance the rate of translation. So the other thing is these mRNA molecules have different half-lives. So some mRNAs will only stick around for minutes and others can stick around for hours. And basically, all of this is a way for me to say that you can't really count on the RNA-seq abundance value reflecting the protein abundance in the cell. In fact, people do these experiments all the time, and the correlation is not as great as you would expect. So you have to sort of keep that in mind in your experiment, that what you're seeing, the dynamics of RNA that you're seeing may not actually reflect some sort of physiologically relevant condition of the cell if the protein abundance actually hasn't changed. So nowadays, I would say there are two major abundance measurements out there. The first is called TPM. It stands for transcripts per million. And it basically says for every million transcripts that could be detected in your RNA-seq library, how many belong to that particular transcript. This is basically the more modern and preferred way of doing RNA-seq abundances, or yeah. And the reason for that is for some complicated mathematical reasons. It is easy to compare these values across multiple experiments, provided that you sequence the same type of RNA. That's kind of confusing because, but I think it makes sense if you sort of think about that the transcripts per million is with respect to the transcripts that are inside your RNA-seq library. So if you do a total RNA-seq, you're going to have lots of different RNAs. Whereas if you do an mRNA-seq, you're going to have less mRNAs. And so your total is actually different in the two cases, so you shouldn't compare their abundance values directly. Okay, there's an older measurement called FPKM, stands for fragments per kilobase per million reads. We generally have sort of moved away from this, but you'll probably see it in lots of papers. It's just a slightly different way of calculating abundance, and it's less comparable across different experiments. So if a bioinformatician ever asks you if you have a preference for abundance, I would suggest you tell them that you would like TPM values. All right, so I wanted to give you guys a sense of like biologically what a TPM might actually mean. So I did a little bit of research, and a couple of groups have sort of attempted to answer the question of basically like, how do you translate a TPM value to sort of a number of copies of mRNA per cell, which is like something biologically interpretable, as opposed to just this random number. So these studies all came to a similar conclusion. It was actually, they were off by like less than an order of magnitude, which in bioinformatics is like a miracle. So they basically said a typical human cell has probably 300,000 mRNA transcripts. So if you work backwards mathematically, that means a TPM value of 3.3 would be approximately a single transcript per cell. I think most people would say one transcript per cell is pretty lowly expressed. The dynamic range of RNA-seq is actually very high. So you can get TPMs that are like very small, like 0.001, and they go all the way up to like in the hundreds of thousands. Yes. I'm going to get to that, but the answer is, in most cases, yes, but not absolutely. The reason is the zero means that of the fragments you sequenced, none of them could be confidently assigned to that transcript. But what if you sequenced 3 times as much from your library, or 4 times as much? If the RNA was present in very, very small amounts, you would eventually detect it. But you have no way of knowing at that point whether you need to sequence more to detect it. It could just simply be absent from your library. But I think I'm going to address that point in a minute a little more clearly. This is my most common misconception. When I first started doing RNA-seq with labs that basically didn't have a lot of experience in genomics, this was often their most common question was basically, well, can you just give me a list of genes that are expressed based on the RNA-seq? And the answer is no, you cannot do that. And there are a lot of different reasons. The first reason is just a biological reason, which is you don't know how many copies of the gene is needed for it to be considered on in a cell. There are genes that maybe do nothing if there's 100 copies in the cell, but if there are 1,000, then suddenly the cell behavior dramatically changes. The other problem is like what your question alluded to, which is for genes that are very, very low abundance, you may have just failed to detect them in the RNA-seq library because you didn't do enough sequencing. And there's really no way to sort of conclusively say that the gene is sort of off or on. All right, so there's another critical problem with this statement, and it has to do with the fact that we used millions of cells to get our RNA-seq library. So imagine that 10% of the cells were highly expressing this gene, and the other 90% had no expression. Your RNA-seq is going to show, some small, moderate expression value after it averages out all those cells, essentially. So then what would you conclude? That it's lowly expressed in all your cells? You'd be very misled. And Tomi is going to talk exactly about this problem in the next talk. Okay, so as I mentioned, comparing two samples is actually the most powerful way to use RNA-seq in my opinion. This is because you control for a lot of things, and you also get to ignore all the stuff that doesn't change between your two conditions, which allows you to throw out a lot of information that would otherwise confuse you. When you do RNA-seq experiments between two groups, the critical ability to detect differences is based on essentially how many replicates you do. If you had to choose between doing more replicates versus sequencing fewer replicates at higher depth, you should always choose to do more replicates. And maybe not always, but 99% of the time, that's the best choice for you to make. The reason for that is because the mathematics behind differential expression are all about detecting expression variability between your samples. And this variability comes from a lot of different sources. Some of it is just biological. The cells are not perfect machines. There's a lot of noise in gene expression. The expression will fluctuate over time for various reasons, some of the reasons we don't even understand or can't control. There is some sampling issue, like I mentioned. If you take a biopsy from somebody and you wait too long before extracting the RNA, the cells will have responded and sort of altered their gene expression profiles. That's going to introduce noise to your replicates. So we often see this, for example, when different technicians do the experiments. So if you have two different people draw blood on the same day from the same person, do the sequencing, you can actually see differences. The other source of variation is technical. We don't believe that the sequencing itself introduces variation. The sequencing seems very stable. If you take the same RNA-seq library, sequence it multiple times on an instrument, you generally get extremely well correlated abundance values. But if you have a different technician make the library, you can get differences. If you do the library generation on a different day, you can see differences. It's a mess, and the best way to combat all these problems is to do as many replicates as you can sort of reasonably afford. Also, it's a good idea to sort of talk to a bioinformatics person before you design your experiment, because people often do batch effects that will confound their results. For example, they'll have the same technician do the blood samples for the disease patients and a different technician do the blood samples for their control patients. It's not a good idea because now you have a confounding variable. Okay. I'm going to give you some examples of some RNA-seq differential expression. So this is actually some data in the lab, but I've sort of anonymized it. In this experiment, we had 4 samples of treated and 3 of untreated. You could pretend this is like a drug treatment, for example. So in this gene, this is an example of what you might sort of naively think is being differentially expressed, but the high variation will cause the RNA-seq differential expression software to basically say this gene cannot conclusively be considered differentially expressed. And that is because these samples within the treatment group are extremely variable. Some of them are actually lower than the untreated, whereas others are much, much higher. So it's going to take all this into account, and it's going to give you basically a p-value that says what's the likelihood that this difference occurred by chance. And in this case, that likelihood was very, very high. You can imagine if you just sort of naively averaged these samples and compared it to the average of these, you would see quite a difference, but that has sort of concealed from you the variation between the samples. So here's another example. This is sort of getting to your point again. So in this case, the detection was basically 0 for the treated samples, but very small for these untreated samples. In fact, it's probably like less than 1 TPM. So if we go by our little mRNA estimate, we would say that on average, this gene is not expressed at more than 1 transcript per cell, which is pretty low. So in that case, what do you think you would include? The software actually found that this was actually quite confident that there was a statistical difference between the expression in these two groups. But as a biologist, you might look at this gene and be like, well, that doesn't make any sense. You know, this gene needs to be expressed in very high abundance to sort of do anything. So this is sort of a great example of sort of what you're going to get out of RNA-seq results. You're going to get results You can't just blindly say differential, not differential. You kind of have to use some knowledge of biology to filter them and sort of decide what you think is realistic based on individual gene knowledge in many cases. So here's a good example of sort of a positive result. So here we have pretty high expression. It's not super consistent among our samples, but it's consistently higher than the other three. And so the RNA-seq software decided that this gene, in fact, was differentially expressed between R2 status. Whoops, sorry about that. So there are a couple other things you can do, which I'm just going to briefly mention. I'm not really going to go into it. When you have a list of differentially expressed genes, you can then sort of study those genes as sort of a set. And you can sort of ask, is there something, do those genes have something in common? And that could be based on pathways. You guys know probably like different biological pathways that are described by the gene ontology database, which allows you to look at thousands, I think, of different genetic or gene pathways. You can also do variant analysis. You could try to sort of correlate genetic variants with these changes in expression, which is very common that a SNP could interfere with the expression of a gene. So maybe your results can be explained just by genetic variation. There's something more advanced, which is basically an attempt to detect sets of genes that are sort of co-regulated in your data. This is called WGCNA. Yeah, I can never get that right. Actually, Tommy would probably be the best person if you had questions about how that works. Okay, so that's it for the design interpretations. Any questions so far? Yes. Yes. Yeah, so generally, let's say you were doing an experiment with cell lines, and you played out 10 copies of a cell line, and 5 get treated, 5 don't. You pretty much want to do all of your library prep, all of your RNA extraction, all of basically everything down to get the RNA-seq library on the same day by the same person. Like that would be ideal. Emily, do you have any thoughts on that?
Can I also add something?
Yes.
I predominantly work in research and I do a lot of technical people, but I think that also you need, if you are going to work with RNA, you need to make the decision of which type of RNA you're going to work with before you do extraction, because we've run into a lot of clinical projects where they've already extracted the RNA and they can't ask the questions that they want to because they've already extracted it. And there's a lot of different kits out there where you'll lose certain parts of RNA, small RNA, you'll lose in extraction. So it's one of those things that is good to get with your bioinformatics team and discuss that before anything is done to the samples.
Yes, I totally agree. Thank you. All right. Anything else? Nope. All right. So I'm just going to briefly describe some resources you guys may find useful if you're beginning some RNA-seq experiments or investigation. So one is the GTEx portal, which is a huge database of RNA-seq experiments. It's like over 10,000 of them currently. These, I think, exclusively come from cadavers. Is that right, Byung-gil? Yeah. They come from cadavers, right? Oh, okay. He's not sure, but so these are tissue-specific experiments of different individuals, and I think in most cases, the genotype of the individuals is also available. So this allows you to sort of investigate whether certain genotypes might be affecting gene expression. There is a similar database called the Expression Atlas. This is not restricted to humans, so it includes a lot of different model organisms, but it's similar sort of tissue-specific gene expression. The NCBI Geo database is essentially the primary repository where published RNA-seq data gets placed. So if you read a paper, more often than not, you'll be able to download their raw data if you want to analyze it alongside yours. And there's also this Tumor Fusions website, which is essentially a good reference database for all the different gene fusions that are have been detected in various cancers. So, all right, that is all I have. Any questions? Oh, yeah, I think you guys will have. They can access these slides, right?
Yeah, everything's going on the website.
Okay. All right. Thank you very much.